participants from across the country, colleagues and dear students, and my dear volunteers. Today, we have our third hands-on session which has two speakers. Our first speaker is Dr. Ruchi, who shall be talking on sequence alignment, and thereafter Dr. Piyush Kumar will deliver his talk on finding evolutionary relationship between different organisms by using software Mega. Sequence alignment is very critical in finding evolutionary relationship between different organisms. I think today's sessions, therefore, is going to be extremely interesting and gripping. It is my honor to introduce my first speaker for the day. Dr. Ruchi is my respected colleagues in Department of Botany, Zakir and Delhi College. She is working as an assistant professor. She completed her bachelor degree in botany in 2000 from University of Delhi. She is alumnus of University of Delhi and attained her doctorate in 2009 with a specialization in molecular genetics. She had published her research work in highly reputed journals like Genetic Resources and Crop Evolution, International Journal of Integrated Biology, and many more. She is also an author of a book chapter in a publication by Springer. She has got behind her teaching experience of approximately 14 years. She has taught different papers of botany and life science till now. However, she had taught paper of bioinformatics at undergraduate level since its inception in 2012. I feel this long association of hers with this paper makes her extremely passionate about this bioinformatics, and this organization of this workshop is her brainchild. Before I introduce my speaker, I request all my students and participants, please write your question in chat box. I now invite Dr. Ruchi to deliver her talk on sequence alignment.
Thank you, Dr. Savinder, for such a warm welcome and my introduction to the participants. So before starting my presentation, I just want to tell to my participants that this particular session, the first session for today, is actually divided in two parts. First, I will be dealing with the theoretical aspects of sequence alignment, and then we'll be doing hands-on session. And for hands-on session, you are already provided with the files that you can do side by side along with me, or you can practice it later on. So first we will be dealing with the theoretical aspect of sequence alignment, and let me share my presentation with you. I hope the presentation is visible to you all.
Yes, ma'am.
Okay. So whenever we talk of sequence alignment, like first of all, what is alignment in general? So we can say alignment is arrangement in a straight line or in correct relative positions. So specifically, when we talk of the sequence alignment, what is it? Sequence alignment is a way of arranging the sequences, maybe DNA, RNA, or protein to identify regions of similarity that may be a consequence of functional, structural, or evolutionary relationship between the sequences. Now, this is the definition of sequence alignment. But if somebody asks you, what are we doing the sequence alignment for? So the first thing that comes in our mind, we are finding similarity amongst the sequences. And actually, what are we doing? We are aligning two or more sequences and we are testing two different hypotheses, either that the alignment has occurred by chance or the two sequences are homologous. Since you know that the DNA sequences are made up of just four base pairs, like everybody knows it's adenine, guanine, cytosine, and thymine. So if I say that there are only four alphabets in our English, there are only four alphabets in English language. So the probability of forming words would be very limited. Same way, all the DNA sequences are just made of these four bases, and because of this limitation, there is a possibility that when we are aligning the sequences, there is, although the sequences are not homologous or the sequences are not belonging to same descent, but we are getting similarity, and that is similarity by chance. So based on this, there is need to assign score to each comparison of a pair of characters, and the scores for all position in an alignment are then added to calculate a total score, which is used to select optimal alignment among the alternative alignments. So we will see later on that we have several tools that are available, although we will be doing only one in today's session. So there are several tools that are available that actually calculate the similarity between the sequences and then gives you an answer in the form of an alignment which is the best suited. But why this similarity is being measured? Similarity measurement is the step to test homology hypothesis. Now this word homology is really very important when we are doing evolutionary studies. And another
Excuse me,
term,
ma'am.
yes,
Uh,
yes, Zishan.
we are still on slide 2. I'm not sure if you are moving it.
No. This is slide 2 only.
Okay, thank you.
Okay, so one more term that you should know, that one is the similarity by chance and the second is statistically significant sequence similarity. Now, what do you mean by statistically significant sequence similarity? So when we have similarity in between 60 to 80% between the pair of sequences, we say that we have got significant sequence similarity, and we are then stating that the sequences are homologous. And what does it mean? It means that homology means that the sequences are diverged from a common ancestor in the past. Now, if you look at the slide 3, these pictures are being taken from EBI website, and these pictures can depict the difference between similarity, homology, and unrelatedness. Like you can see in the first picture, there is a starfish that has been compared to a man dressed up like a starfish. In the second picture, you can see a chimpanzee working on a typewriter and a man working over the computer because they have a common ancestor. So this is homology. And the third picture that you can see on your screen, it is totally unrelated. So now when we talk of homology, it is and means important term. It's a notion of sameness formalized in concept of homology, and we infer homology when two sequences or structure share more similarity that would be expected by chance, and when the significant similarity is observed, the simplest explanation is that the sequences did not arise independently and they arose from a common ancestor. So, with reference to protein or DNA sequences, homology simply means that the given sequences, they share common ancestry. And with respect to homologous sequences, there are two types of sequences. One is the orthologous sequences. Now, orthologous sequences, they have evolved from common ancestral gene by speciation, but they have retained performing the same function in the course of evolution. Like, for example, the sequences of human alpha globin and mouse alpha globin. Although the sequences are present in different organisms, that is human and mouse, but they perform same function. But when we talk of paralogous sequences, the paralogous sequences, they actually arise due to a gene duplication event within a genome. And these genes, they perform different or new function even if these are related to the original one. For example, the human alpha globin and human beta globin gene, although the genes, they are present in the same organism, but they are performing different function. Now, so on the basis of the discussion, we should understand that by doing alignment, we are calculating the similarity and we are making a hypothesis that the similarity is by chance or it is significant similarity. And if the similarity is significant, then we say the sequences are homologous. So homology is an inference or a conclusion that we draw based on the observed similarity. And the sequence regions that are homologous may be called as either conserved, consensus, or canonical sequences. There is a term that is often used that is percent homology. It's a misnomer. There is no term like percent homology. It is the percent similarity on the basis of which after a significant value, we can say the sequences are homologous or not. Although in case of specially protein sequences, we can observe partial homology. And what is partial homology? Where a segment of compared sequences has shared origin while rest does not. And as I said, this can be observed especially in case of protein sequences, that when we are comparing two protein sequence, a fraction of sequence is dissimilar in the two sequences, whereas a segment of sequence can be same and it depicts common descent. So partial homology can occur where a segment of compared sequence has shared origin while rest does not. Now, again, the question, why sequence alignment? So as I told you in the beginning of the talk, that actually when somebody asks you, why are we doing sequence alignment? So the first thing that comes in our mind, because we want to detect the similarity between the sequences. And also the sequence alignment is used to detect conserved regions or motifs in the sequences, especially in case of protein sequences. And also the sequence alignment is useful for detection of structural homologies like pattern of hydrophobicity or hydrophilicity, gaps, and thus it assists in improved prediction of secondary and tertiary structure of proteins. Then sequence alignment is also helpful in prediction of features of aligned sequences like conserved position, which may have structural or functional importance. Then you will see in the today's lecture itself, in the second lecture, that sequence alignment is a prerequisite for inferring phylogenetic relationships. So whenever we need to construct a dendrogram, phylogram, cladogram, or we have to study phylogenetic relationship by using DNA or protein sequences, First, we need to do sequence alignment, and most of the programs that are available nowadays, we will be using Mega X. They have inbuilt sequence alignment program. So first, the program that is used for construction of phylogenetic tree, you will be seeing later in the day. First, they calculate similarity or sequence alignment and then use that data to predict the phylogenetic tree. Now, whether we are comparing two or we are aligning two or more than two sequences, the concept of alignment remains the same. And whenever we are doing bioinformatics, especially keeping in mind construction of phylogenetic tree that we are doing today. So we are assuming that the sequences, they share common ancestry, and common ancestry, that means the sequences that we are undertaking in today's investigation, they have originated from a common ancestor. So kindly look at the figure on your screen. So the first part of the picture, you can see the common ancestor, and it is a DNA sequence in, as I told you earlier also, that ATGC, made of ATGC bases, and each A, T, G, and C has given a different color code. Now, from that common ancestor, we have two descendants. Okay, and then what we have done is, in the third part of the picture, we have aligned them. Now, to compare two or more sequences, it is necessary to align conserved as well as non-conserved residues across all the sequences. And when these sequences are aligned, then only it is possible to identify the locations of insertion and deletion in the descendants, since their divergence from their common ancestor. Now, look at the aligned sequences in the figure. So, first two position T and A, and there is a perfect match. In the second sequence also, we have T and A. But at the third position, we have, in the first descendant, we have a, whereas in the second position, second sequence, we have a space. Then again, there is a perfect match till the position 9, and at position 9 we have, in the first sequence we have the T, whereas in the second sequence we have G, and this is mismatch. Then again at 12th position there is a C in the first sequence, whereas a space in the second sequence. So whenever we are doing alignment, there are three possibilities. One is the bases match, the bases mismatch, and there is base in one sequence, but no base in another. Coming back to the figure, you can see that at the third position, either there is insertion, so there is insertion in descendant 1 that has led to a space in the sequence 2, whereas at the ninth position there is mutation in descendant 2 that has resulted in a mismatch, whereas again at the 12th position we have deletion in descendant 2. So what we have observed, after alignment we have matches, mismatches, and then we have spaces, and these are the central elements to calculate score of an alignment. Now, so what we have observed, match, mismatch, and gaps. The next thing that comes, so if there is no mutation in the descendant, okay, then the sequence, original sequence, and there will not be any difference between the original and the descendant. Actually, what is a gap? Gap is a maximal consecutive run of spaces in a single sequences of a given alignment, and gaps are actually inserted because they help to create meaningful alignment. The idea is to treat a gap as a whole rather than give each of its spaces the same weight. There are many ways in which weight can be given to a gap. The concept of gap in an alignment is important in many biological applications, since the insertion or deletion of entire subsequence often occurs as a single mutational event. Moreover, many of these single mutational events can create gaps of varying size, and we will need to score a gap as a whole when we are trying to align two sequences of DNA, so that we should not give high weightage to a mutation, to a single mutation, because of which maybe many gaps of variable lengths are created. At the protein level, two protein sequences might be relatively similar over several intervals, but they differ in interval where one contains a protein subunit which is not present in other. Again, the introduction of gap will help us treat these cases as good matches. So basically, what is the relevance of gap creation? So as I told you, if there is no mutation in the original sequence, there will not be formation of descendant sequences. So sequences, they have diverged from a common ancestor through various type of mutations. First is substitution. As you can see, the sequence ACGA after substitution has changed to AGGA, or there can be insertion in which the 4 base pair sequence ACGA turned into ACGGA, or there can be deletion in which a 4 base pair sequence ACGA can change to AGA, and the last two, insertions and deletions, will result in gaps in the alignment. When we say substitution results in mismatch, whereas insertion or deletion, many times indels they are called, they are result from insertions or deletions by the insertion and deletion mutations. Now, whenever there is gap in the alignment, what does it mean? It means that the alignment is not perfect and there the gaps should be penalized. That means we cannot get a alignment score of 100% similarity. So for that, the gap penalties are used in sequence alignment to penalize the gap. And the gap penalties actually contribute to overall score of alignment. When we talk of gap penalties, the gap penalties are often composed of two parts. One is gap opening penalty and second is gap extension penalty. And by convention, the gap opening penalty is denoted by D, whereas the gap extension penalty is by E. And always the gap extension penalty, the value of gap extension penalty is kept very small in comparison to gap opening penalty. So gap, why is it so? Why gap extension penalty is usually smaller than gap opening penalty? As you can understand from this example, 10 insertion of one nucleotide each should be harder than one insertion of 10 nucleotide. That is, the gap opening is less probable than a single gap extending over more than one nucleotide. And because any type of mutation can result in gap, any type of mutation like insertion deletion can result in the gap is a rare event also. So when we say we have 10 different gaps at 10, we have gap of 10 nucleotides at different 10 places, it is much harder in comparison to a single stretch of gap at one place, 10 nucleotide at one place. Now, when we talk of gap penalties, the gap penalties are of two types, linear gap penalty and affine gap penalty. Now, linear gap penalty is the simplest type of gap penalty and it is always negative. And what linear gap penalty consider is, in case of linear gap penalty, whether although we have discussed that 10 nucleotides at the gap, 10 gaps of one nucleotide each and at 10 places are harder than a single gap of 10 nucleotide at a one place. However, in case of linear gap penalty, the overall penalty of 10 gaps at different places and at the one place is kept same. So it actually does not give us a true picture of correct picture of alignment score. So to overcome this problem, there was introduction of affine gap penalties. And in case of affine gap penalties, it is much more likely that one big gap of length 10 occurs in one sequence because of a single insertion or deletion event than it is for 10 small gaps of length 1. Therefore, affine gap penalties have a gap opening penalty and a gap extension penalty. And as earlier also I mentioned that the value of gap opening penalty will always be lesser than gap extension penalty. Now, there can be different types of alignment on the basis of different criteria, like on the basis of whether we are comparing whole sequence or we are comparing a part of the sequence. There are two types of alignment. One is global alignment and second is the local alignment. Now, what is global alignment? First of all, global alignment. So global alignment, what it does is it compares sequence as a whole. So if we are comparing two sequences, the whole sequence is being compared from 5' end to the 5' end to 3' end of one sequence is complete, means the whole sequence is compared from the 5' end to the 3' end. So when we talk of global alignment, it compares sequences and it gives best overall alignment and will return only the best matching segment for a given pair of sequences. But it may fail to find best local region of similarity among the distantly related sequences. So in comparison to, we will later on comparison with local alignment, we will deal with global alignment in bit detail with an example. So when we talk of local alignment, actually local alignment compares only the identical or similar sequence patch in any part of the complete sequence. So when we talk of local alignment, it finds region of ungapped sequence with high degree of similarity. Local alignment is better at finding motifs, especially for sequences that are different overall, and it can return more than one matching segment for a given pair of sequences. Now kindly look at the figures that are there on your screen. So in case of global alignment, I'm sorry. So you can see in case of global alignment, the two sequences, one is target and the second is the query sequence, they are compared wholly or at the whole length. Whereas in case of local alignment, only a particular part within the sequence is compared. So in case of global alignment, it is assumed that the two sequences that we are comparing are basically similar over the entire length of one another, and the alignment, the global alignment, it attempts to match them to each other from end to end, even though the part of alignment are not very convincing. Or you can say that in global sequence comparison, the algorithm seeks to align every residue in one sequence with every residue in a second. In contrast to more commonly used local sequence alignment algorithm, which seeks only strongest region of similarity between the two sequences. Then if you see the local alignment, it searches for segment of two sequences that match well, and there is no attempt to force the entire sequence into an alignment, just the parts that appear to have good similarity according to some criteria. Using the same sequence that we have seen in case of global alignment, you can see that the local alignment only finds the best match within the sequence, and it actually gives you the localized sequence match. So this is the difference between global and local alignment. However, the when we are performing the alignment with different motifs, we have to select either global or local alignment according to our needs, that what we actually do want to do after the alignment, whether we want to study the phylogenetic relationship or we want to study the particular location of a particular motifs and so on. Now, as we have seen that on the basis of what part of the sequences are aligned, we have two types of alignments, global and local. However, when we are aligning, when we are means on the basis of number of sequences, and when we are aligning only two sequences, that is pairwise alignment. And when we are aligning more than two, three or more than three sequences, then it is multiple sequence alignment. And later on, we will be studying the multiple sequence alignment, using how to do multiple sequence alignment using the tool Cluster Omega. Now, the dot matrix method is one of the easiest method that we can use to study or to align a pair of sequence. So what is actually a dot matrix analysis, or how the dot plots are made? So a dot matrix analysis, it's a method for comparing only two sequences, and it is the way by which we can look at the possible alignment. What is done in dot matrix analysis? One sequence A is listed across the top of the matrix and the other B is listed down the left side. And starting from the first character in B, one moves across the page, keeping in the first row and placing a dot in many columns where the character in A is the same. And this process is continued until all possible comparison between A and B are made, and any region of similarity is revealed by a diagonal row of dots, and isolated dots that are generated which are not on the diagonal, it represents the random matches. Now, this figure on your screen gives you the simplest example of a dot plot. So what we have done in this is we have two sequences, sequence 1 and sequence 2. And you can see what we have done is we have compared the sequence 1 with the sequence 2 in a checkerboard. And wherever there is a match, we have placed an asterisk. And then after all the asterisk position, what we have done is we have drawn the diagonals, and you can see within the diagonal position there are some, within the diagonal, three diagonal segments, there are certain gaps also. So these diagonals actually they represent the alignment, whereas the gap in the alignment, the gap in the diagonal, it shows the mismatches. So you can see we have compared 1 and sequence 2, and we have observed the diagonals that denotes the matches among the sequence or the alignment. Now, the figure that I have shown you, it is showing the comparison of the nucleotides of few bases. However, the figure depicted on these particular two pictures, these are representing when we compare the sequences which are much larger in size. So first of all, whenever we are comparing the sequences, two sequences, we are getting what is dot plot. Now, what is dot plot? Dot plots are two-dimensional graphs showing the comparison of two sequences. The two axes of the graph represent two sequences that are being compared, and every region of the sequence is compared to every region of the other sequence. Now, kindly look at the picture on the right hand side corner above. So you can see that we have observed the diagonals also, and within the diagonals you can see the diagonals are connected by a horizontal line, right? And actually, just looking at the dot plot, we can make out that this horizontal line indicates or represent either an insertion in sequence 1 or deletion in sequence 2. Whereas if you look at the picture lower to it, we have, in addition to the diagonals, we have vertical lines as well. And these vertical line, when we are combining the two diagonals that we have got, the vertical line, either it represent deletion in sequence 1 or insertion in sequence 2. And of course, as I told you, that the diagonal, it represents a perfect match. And so by only looking at the dot plots, we can make out that the two sequences along the length showing alignment between which base pair to which base pair, then insertion or deletion in the sequences in which part, and then again matches or mismatches in the other parts. So when we interpret the dot matrices, the regions of similarity, it appears as the diagonal runs of dots. The reverse diagonals, that is the line perpendicular to the diagonal, it indicates inversions, whereas the reverse diagonals crossing the diagonal, that is making a cross, indicates palindrome. The situation will be more clear when you observe this particular picture. So you can see in the figure, in the sub-figure A, only one diagonal can be seen that represent a perfect match. Then, in addition to a one central diagonal, there are two more diagonals that can be observed in the sub-figure B, and those represents the repeats. Whereas in the figure C, we can see palindromes, in D, partial palindromes, in E, from the dot plots, we can predict the presence of microsatellites, then mini satellites, then homology and indels. So just by looking at the dot plots between the two sequences, we can see whether there are presence of inverted repeats within the sequence, or palindromes within the sequence, or micro or mini satellites within the sequence. Now, coming to the last part of the lecture, theoretical lecture, that is multiple sequence alignment. Now, when we talk of multiple sequence alignment, the multiple sequence alignment, as I told you, when we are aligning more than three sequences, this is multiple sequence alignment. And the multiple sequence alignment is the basic tool for sequence analysis, and it is used to identify highly conserved or similarity regions within a set of related sequence. There are several factors which affect the analysis of conserved region in the set of sequences, such as number of sequences included in the analysis and the ratio of number of almost identical sequences to the number of sequences that are more distantly related. Now, whenever we are talking about multiple sequence alignment, the concept remains the same. And in multiple sequence alignment also, the first, irrespective of how many sequences, although the result is affected by the sequences undertaken in the study. But then also, first one sequence is compared to the second sequence, then both the sequence, the best match sequence, are considered as one, and then they are compared to the rest of the sequences. Then it is like merging one sequence with the other until and unless all the sequences that we are undertaking taken into taken into account. Then the multiple sequence alignment. So why do we use multiple sequence alignment? Again, the multiple sequence alignment is also useful for detecting similarities between the sequences, whether the sequences are closely or they are distantly related. And also the multiple sequence analysis has role in functional prediction. Like, what is the meaning of functional prediction? Functional prediction means that when we are comparing a protein sequence with the unknown function with another protein sequence with the known function. So if we find significant similarity or homology, then we can predict that maybe the protein sequence with the unknown function is performing same function as the protein to which it is compared with. Then again, the multiple sequence analysis is also used for studying or detection of structural homology that we have mentioned earlier also. Then it is the basic tool for construction of phylogeny that you will be seeing later on. And whenever we want to distinguish between orthologs and paralogs, as I mentioned in the beginning of the session, that when we are saying paralogs, the paralogs are actually arising from the gene duplication event. So when we have to distinguish between orthologs and paralogs, we have to do multiple sequence alignment. Now, coming to the hands-on session. So if participants, you want to do along with me, what you want to do is you can open your Safari or your Google, and you can type this. Just a moment, please. And you can type this URL, and this URL will lead to this particular page that is of Cluster Omega. So for hands-on session, we will be using the tool Cluster Omega, and we will be studying the multiple sequence alignment through Cluster Omega, how actually we do it. So in the yesterday's lecture, let me share my screen again with you. So Dr. Zishan, is my screen is visible?
Yes.
Yes. Okay. So if you type that URL, you will come to this page of Cluster Omega, and the Cluster Omega is a tool from EBI, that is European Bioinformatics Information. It's a tool that we can use for detecting multiple sequence alignment. So if you see, it is such a easy tool in which we have actually only 3 steps. So whenever we are doing multiple sequence alignment, so we just need this Cluster Omega and the sequences as mentioned by yesterday's speaker in the FASTA format on a notepad file. And then we can easily perform this multiple sequence alignment. So you can see that the first step is we need to enter our input sequence. So the sequences can be entered either in the form of protein or DNA or RNA. So first we will do the multiple sequence alignment of protein sequences. So either what you can do is you can just paste the sequences, faster format sequence from the notepad, and you can just copy the sequences in the faster format from a notepad and paste it in this blank. Or what you can do is just click on the choose file and then directly upload the sequence from your computer. Okay, just wait for a moment, please. So what we have done is the screen is visible.
Yes, ma'am.
Yes. Okay. So what we have done is from our notepad file, we have copied the sequences, the protein sequences, and then we have the after copying the sequences, we have set your parameters. Usually for beginners, we don't fiddle with this parameter section much, and this default setting that is provided by EMBL or EBI, it fulfills the need of most users. So there is no need of changing this particular section, and then you have to just click on the submit. Now, if we want these results on our email, so what we can do is we can just click over this and we can write our email, and by what file we want to have this, by what name we want to have this file in our email, and then we can just click over the submit button. But because for now I don't want these results to be notified in my email, so I am simply clicking over the submit button. Just a moment, please. Dr. Zishan, what is visible on the screen? Is the alignment
Yes,
has come?
yes, ma'am. It's visible.
Okay.
Uh, it's better if you zoom a little bit. So
Okay, I will. But actually, zooming in this case will not help. I will zoom it little later. First, I just want to explain something. So the you can see over my screen that these are the results that I have got. Okay, and you can see that you can see my arrow cursor. So it is over the alignment, and by clicking at different sections here, we can get different information about the sequence alignment, like result summary. We can also observe the guided tree that is made by this alignment. We can observe the phylogenetic tree as well. And however, we are not interested right now in this. We are just doing alignment for now. So I just want to explain you something. Now, the second thing, first is alignment, and second is, you can see at the end, that is the show colors. So if I click on show colors, you can see that all of us knows that the protein are made of 20 amino acids. So when I click on show colors, each one of the 20 amino acid has given a particular color code, and we can observe just by looking at the alignment where red is coming, means in a particular alignment where we are seeing that only red color is coming. So just by looking at it, we can see that there is perfect alignment. Now, if you come to the alignment part, one thing that I want to tell you is that alignment in the beginning and at the end is usually having more gaps, and whenever we are analyzing the alignment, we actually negate these parts and we consider the parts that are in between. And then, so what we have done is, in this particular alignment, I have zoomed. So what we have done is we have aligned the 10 sequences, and you can see that the alignment can be seen in three parts. And just after the alignment, you can see the presence of asterisk, the presence of colon, the presence of a blank space, and the presence of a dot. Now, by looking at this asterisk, colon, and dot, these are the positions that give you some indication about the alignment at this particular position. Okay, so the asterisk, it actually indicates perfect alignment, like the 10 sequences that we have compared. In all of the 10 sequences, we have same amino acid at this particular position, whereas the colon, it indicates a site belonging to group of exhibiting strong similarity, whereas a dot, it indicates a site belonging to a group exhibiting weak similarity. And where there is a blank, those areas actually represents the hypervariable region in the alignment. So what we have done is, in clusteral omega, so we have just pasted our protein sequences, 10 in number, in the search box, and then we have clicked on the submit, and we have got the alignment in this form, and we can just click over the color, the show colors icon, and we can get color coding. Otherwise, in black and white, we can get the alignment. Same thing can be done for the DNA sequences. But in that case, instead of the protein sequences, we have to choose the DNA from the scroll down menu, we have to choose DNA, and then we have to choose a file in which the sequences, I don't know for what reason this option of choose file is not working. So we need to go back and we need to copy the sequences. And again, after pasting the sequences, DNA sequences, we have to click on the submit icon. Okay, let me do it once again. Just wait. Okay, now can you see the screen?
Yes, Dr. Ruchi.
Okay. Okay. So now you can see this alignment is achieved by using the DNA sequences, and the icons that are appearing, that was appearing for protein and the DNA sequences are the same. And alignment, result summary, guide tree, phylogenetic tree, result viewer, submission details, and also I don't know for what reasons show color is not coming, but otherwise maybe of connectivity issue or something like that. Otherwise, show color icon also comes, and ATGC has are given a different color code, each base with the different color code, and then we can just observe then we can just observe the alignment by looking, means by just observing the alignment we can find the regions of similarity, we can find the variable region, hypervariable region, and so on, as I explained in case of protein sequences also. So I hope this is clear to you, the hands-on as well as the theoretical part of sequence analysis, sequence alignment. And I just want to thank everybody who attended this session. And I hope that this session was fruitful for you. And thank you very much. Thank you, everyone.